What's up, everybody? So this was going to be kind of a lot, but not a lot, but kind of a lot, but I'll go through and explain it and kind of like normal. So this is actually going to get into how psychology is a science and all about research methods, research methodologies. So here we go. All right. Why is it important to use scientific guidelines for psychological research? You're going to answer the question. All right. So words to remember. You're not going to need to write these down, but this is just good to know, right? Objective things, observable, able to be seen, heard, touched, counted, described, right? We can all see the same thing, and it's as close as fact as we can get, right? Because if it's I saw, I counted, this is what they did, right? They did this action, whatever it may be, right? It's objective because we can see it. We're not making judgments about anything. If we start to make judgments about why a behavior occurred, now we're getting into the subjective, right? Subjective information, opinions, beliefs, there's the judgments, assumptions, whatever it may be. The big issue is it's biased. Once you get into biased takes, it's no longer objective and it's no longer good, strong, viable data, right? So if it gets into they didn't want to do this, or they don't like that, or they were feeling this, or they thought that, you don't truly know that. That's you kind of assuming and guessing what the reasons behind an action were, and that's subjective. We always want to be as objective as possible. So, selecting participants. Random selection is from a specific population to find participants. Now, here's what that means. The population, this is in regards to when you're conducting an experiment, the population is like, who are you choosing from? So if I were to poll and give a survey to all of the students at Spelman, the population would be the student body of Spelman, so all 500 whatever amount of kids here. That would be the population. Random selection is exactly what it sounds like. Randomly getting selected to take part in the survey. So everyone gets a number, 1 through 550 or whatever it is, and then 100 people get pulled out of a random number generator. Randomly selected. Random assignment, again, exactly what it sounds like. So let's say I want to study 100 kids at Spelman, and I have a group of 50 and a group of 50. Randomly, people get assigned into each one, right? There's no reason as to why a student is in group A or group B, right? It would just be totally random. The experimental group, the group exposed to the independent variable, right? The one that's truly being experimented on. And then the control group, hopefully these two things sound familiar to you based off science class. Your control group is the one that should serve as the steady thing that if there's a change in the experimental group, that way you can say like, oh, here's the difference, right? So use control the possibility of other factors. You guys know controls. You've been through science class. Variables. I'm not going to really go through independent, dependent. I'm really hoping you guys know this by now as seniors. But, right, it's a thing that is being changed, a thing that's being manipulated. The thing that will cause the change. The dependent variable, right, is dependent on this guy, the independent. Right, so dependent is dependent on that. So this is the group being experimented on or not experimented on. Dependent would be what is the difference between the two. The big part is confounding variables. So confounding variables is something other than the independent variable that might produce an effect on the experiment and skew the results. What that means is, let's say there was a medical experiment on a new ADHD drug. The independent variable would be is if the group of people in the study got the drug or the experimental drug or like the sugar pill, right? Independent, which pill are they getting? The dependent would then be like what obviously the impact it has. A confounding variable, and something that is actually seen a lot in medical things is interesting, like the diet of the person could actually impact the effectiveness of the new drug. For example, there's actually a lot of pills where like one of the things that says like you can't eat grapefruit because the acidity of the grapefruit actually impacts the like whatever pill that you're taking, whether it's for your heart or something like that. Like that grapefruit would be a confounding variable because it's something that is actually changing the outcome of the study, but it's not supposed to be there. It's not supposed to be like impacting something. So confounding variables are something that can mess with results that are not supposed to be there. Randomization, so like random assignment, random selection, stuff like that, is supposed to help limit the effect of confounding variables. Possible problems in research. The Hawthorne effect, also known as like the observer, right? Behavior of participants may change as a result purely of being in the experiment. Simple terms. When people know they are being watched, they act differently. Right? When it's just you guys and me in the room, right? We have our own culture and way that we interact. When Hodes or Kelly or an authority figure or someone from the outside walks into the room, people, right? Hopefully you guys act differently and maybe sit up a little bit straighter, raise your hands, all that good stuff, right? But people act differently when they know they're being watched. The placebo effect, right? Results caused by expectation alone show changes dependent variable only when given a placebo. I'm assuming you guys know what a placebo is, but it's basically you're given, you're in a medical study, but you're given the sugar pill. But you say like, oh my God, yeah, this is really helping. No, your brain is just tricking you. And lastly, the experimenter effect. The behavior, expectations, and biases of the experimenter can cause a participant to change his or her response. What that means is, like in a medical study, if you were part of it and I was the doctor, and it was, whatever the medication was for, if I walked in and just like threw the pill at you and like generally projected that I didn't care about being there and I didn't care whether it worked for you or not, that was going to impact you and be like, oh, I'm not getting the real thing, so this is useless. Versus if I came in and like carefully gave you the pill and took really detailed notes about how you're feeling and how you're reacting to everything, you would start to think of, because my behavior is saying like, oh, I'm actually getting the real medicine. So the way that the experimenter, like the scientist, behaves can impact your interpretation of the entire study. A way to get rid of some of these things, Hawthorne, placebo, experimenter, is with single blind or double blind studies. The best thing is always the double blind. So in the single blind, the participants don't know what they're getting. I think like in experiments, but the doctors do. In a double blind study, neither the participant or like essentially like the people giving the medicine know who's getting the sugar pill or the real like tested medicine. So that way their behavior can't impact the person receiving that pill. This is going to come up throughout the year, just so you guys get this. But if data is valid, that means it measures what it's supposed to measure. And we'll have some stuff tomorrow to help with this. But valid means it measures what it's supposed to measure. The next phrase you're probably going to hate me for by the end of the year because I just say it constantly. But statistical significance means that the results are not due to chance. So again, statistical significance, the results are not due to chance. We'll go further in that in class today, depending, I guess, how late you're doing this. All right, why do we need science and psychology? Hindsight bias, right? Oh, I knew it all along, right? Sometimes it's called like common sense. Oh, of course that would have happened. Well, you don't really know that. Overconfidence. Humans tend to think we are like, we know more than we do, or like we're just better at certain things. It's just trying to like maybe like bring us back down to earth a little bit. Something our brains love to do is perceive order in random events. So like if you're flipping a coin 100 times and all of a sudden you hit heads 5 times in a row, your brain's like, oh, sick, I'm on a hot streak. I'm hitting heads. It's totally random every flip, but your brain loves order. Your brain loves to feel like it's in control. So the world is random. Our brains don't like that. So our brain goes out of their way to like find order because it makes our brains feel better. And then confirmation bias. This is one we've all experienced. Confirmation bias is simply people only search out information that is going to verify their preconceived notions. If they already have a feeling about a topic, they're only going to go find out information that proves that theory correct. And anything that goes against what they believe, they ignore, they say is wrong, is lying, is not real. So confirmation bias, that's something that we especially see modern day in politics. And that's not a one-side thing. That's both sides things, to be very honest, is the unwillingness to view it from the other side and just automatically say, no, that's wrong, that's not true. All right, so these 2 videos are going to be in the PowerPoint for you guys. So they'll be linked in there for you, but that just helps to show, again, some different reasons of how psychology is used and why it's used. So I'm going to end this video here because it's about the 13-minute mark, and then there'll be a second one with just the end of this. All right, thanks.